No,
and
it was-
honoring.
I know, I look out, I see many of you that I know. I'm Nancy Nielsen, currently Senior Associate Dean for Health Policy. God knows what happens after that, we'll find out. I'm a proud alum of this school. Today's program has a few very exciting events. In a few minutes, we're going to hear from Dr. Weissman, our distinguished speaker, who will talk about how he and his collaborators invented and developed the modified mRNA technology that is the basis of the two, I would say, most successful COVID vaccines. His talk will be followed by a Q&A session with a panel of faculty experts. After that, we will introduce this year's distinguished alumni and formally conclude the year-long celebration of our school's 175th anniversary. And those of you who are participating in a class reunion, we welcome you in particular, or if you're here to tour the school, we welcome you in particular. So before we get started, I'd like to acknowledge several groups and special guests here with us today. Thanks to their leadership, the schools and universities partners played a role in today's program. A warm welcome to the many alumni who are in town celebrating a reunion, especially our golden alumni classes of 72 and 71, who are celebrating 50 years or more, because we couldn't do it last year. That's the reason, so just so you know. And the reunion committee who made today's activities possible. So congratulations to the Golden Alumni classes. I also would like to acknowledge and ask to stand Dr. Michael Kane and his wife Peggy, if she's here. We are grateful for his tenure as 15 years as dean, and please allow me to acknowledge him and join me because he brought this school to fruition. We also welcome alumni who have been or will be honored as the Distinguished Alumni Award recipients, including the school's 2022 recipients, Doctors Norma Nowak, Robert Gore, Jessica Reynolds, and Kenneth Gales, as well as all who were honored from our various schools' different events this week. Finally, I would like to particularly thank Dr. Howard Walensky, his daughter Sharon Taffet, and members of the Medical Emeritus Faculty Society for their efforts in bringing Dr. Drew Weissman to Buffalo. As I said to him last night, today is Buffalo, next year it's Geneva. Dr. Brich, I want now to introduce our new vice president. What new, after 6 months, how long do we stop saying new? I'm not sure. Our new Vice President for Health Sciences and Dean of the Jacobs School, Dr. Allison Brashear. She will introduce our keynote speaker, Dr. Weissman. Dr. Brashear is an accomplished neurologist and a leader in academic medicine. We stole her from the University of California at that little place called Davis, where she was dean, so we are very pleased that she came here. Since coming in December, it seems as though she and her husband, Cliff, have met half of Buffalo. We believe that they have, and they're integrated very tightly into the fabric of the city. And we're really very fortunate to have her here as our dean. Welcome, Allison.
Thank you so much, Dr. Nielsen. Good afternoon to our alumni, students, donors, faculty, staff, and community, and family members and distinguished guests. It's so exciting to welcome you here today. Our audience is comprised of medical alumni from 16 different states. While we welcomed our first classes here in January of 2018, this may be the first time, as Nancy noted, that many of you have seen this new state-of-the-art medical school building. Did you know that the graduating class of 2022 was the first class to complete all 4 years of their medical education in this new building. I'm pleased also to share that more than 1/4 of the graduating class, 51 students, will do their residency here at UB. So speaking of medical school classes, I also want to take a moment to extend special thanks to the class of 1971 who have joined us for their 50th anniversary year. This is thanks to their efforts. We're celebrating a new learning landscape on the 7th floor this weekend, and you can see that when you join us on the tour later this evening. Also celebrating their 50th year this year is the class of 1972. And we're grateful that this very busy group have been champions, and they raised the most of our classes in the reunion giving this year. I'm also pleased to welcome Saul Messenger, celebrating 65th reunion this year. Our Active Learning Center, located on the first floor, just one floor down, is named for Dr. Messenger. And like Dr. Nielsen said, I also want to thank Dr. Michael Kane for his vision in getting this extraordinary building built for our students for many years to come. And now the reason we're all here today. I'm pleased to begin our Harrington lecture featuring Dr. Drew Weissman and our 2022 Distinguished Alumni Awardees, a special event that ends a year-long celebration celebrating our school's 175th anniversary. This is my first alumni weekend and first Harrington lecture. I want to thank all of the alumni who have shared their memories of UB and their aspirations for the future of the Jacobs School of Medicine. For over 80 years, this event has drawn proud alumni from all over the country while serving as a forum for prominent speakers like Dr. Weissman. While we have many things to celebrate today, we also gather with heavy hearts in the aftermath of the horrific events that transpired in our city last month. The Jacobs School is committed to fighting hate and racism in our community and in our country. Yesterday, our school was lit up in orange to support the movement to end gun violence. The events of May 14th, 2022 have traumatized the city of Buffalo and left a permanent scar in our hearts that occurred in our community. Buffalo is forever changed by this act of hatred. I am heartened by the countless stories of our community coming together to support those most in need. As part of our ongoing commitment to support the community, the Jacobs School has established a multidisciplinary community response working group that already has many projects underway. The Jacobs School is committed to helping lead enduring change to support those most in need in the city of Buffalo. Now, it is my pleasure to welcome and introduce the 2022 Harrington lecturer, Dr. Drew Weissman, and welcome his wife, Dr. Ellen Weissman. Dr. Weissman is the Roberts Family Professor in Vaccine Research at the Perelman School of Medicine at the University of Pennsylvania. He is best known for his collaborations for RNA biology and COVID-19 vaccines. He and his collaborator, Dr. Caitlin Carrico, invented the modified mRNA technology being used in the Pfizer, BioNTech, and Moderna vaccines to help prevent COVID-19. More than 15 years ago, Drs. Weissman and Carrico found a way to modify mRNA and later developed a delivery technique to package the mRNA in fat droplets called lipid nanoparticles. This endured as it could reach the proper parts of the body and trigger the immune system to fight disease. These laboratory breakthroughs made mRNA safe, effective, and practical for use as a vaccine against COVID. Dr. Weissman and his team are currently working on a pan-coronavirus vaccine to help stop the coronavirus epidemic, an universal flu vaccine, and a vaccine to prevent herpes. His lecture today, aptly entitled Collaboration Caught Fire, Dr. Weissman will describe the highs and lows of the research journey that took over 2 decades and culminated in helping lay the scientific foundation for the largest global vaccine campaign in history. In 2021, Dr. Weissman was the recipient of the prestigious Lasker-DeBakey Clinical Medical Research Award, and in 2022, the Breakthrough Prize in Life Sciences, the world's largest science prize, among many other international awards. The story of how Dr. Weissman and his collaborators maintained their focus and determination against formidable trials and tribulations, ultimately proven right, is truly inspirational, both for our faculty, our students, but everyone across the world. It's also inspirational in how they struggled in obscurity for 2 decades, so much so that many people today don't know the name Drew Weissman or his incredible accomplishments. So before having Dr. Weissman speak, we have a video that we had some fun putting together just to let you know that some people are beginning to know his name. His work has saved millions of lives around the world. And to let you know that his team is still continuing to tackle some of the most complex diseases. So it's my pleasure to introduce you to a video portrait that asks the question, who is Drew Weissman?
Do you know who he is?
I actually do not.
Have you ever heard of Drew Weissman?
I haven't actually.
Okay.
Actually, I do not know who he is.
I don't know, sorry.
At UPenn, yeah, I know him.
No, never.
He studies RNA.
I would have read about his work, but I might not know the name. Everybody wants to talk to Drew Weissman right now.
Yeah, that's very actually exciting.
Wow. Yeah, well, he's basically behind the mRNA revolution, so that's really exciting.
So I am so excited and honored that Drew Weissman is coming to UB.
That's thrilling.
That's awesome.
Oh, it's such a great tale, right? And it's belief in yourself and perseverance is critically important in science.
We're going to do this and we're going to make it happen. And then it goes completely left sometimes.
Even though like everyone was telling him it wouldn't work, and be able to prove other people wrong.
It'd be cool to hear it directly from someone who had a part in it.
I think as a grad student, that would be very encouraging to listen to.
Somebody who has kind of worked in the trenches, a little bit unknown, and now all of a sudden their really interesting, basic, fundamental research became super important.
That's really inspiring to me as a junior scientist.
To hear that he muddled through, that they muddled through, that same sense of we have something great. Oh wait, no one else thinks we have something great.
Yeah, the joke is always that science is just a massive group project.
You have an idea of what it can do, but you don't always know everything where it's going to go.
That's why we call it research. You search again and again and again.
It doesn't always have the answers, but what's beautiful about it is that we can just keep searching for them.
Many times this is what leads to breakthroughs and great discovery.
With this vaccine, there's so much misinformation and so much distrust. So I think it would be very, very beneficial for the public.
Dr. Weissman's group was involved with one of the 3 critical steps. I'd love to hear about his moments of doubt and how he was able to overcome that.
They saved 1 billion lives.
So thank you very much. That was a wonderful introduction. And the video was really great. I always joke, I have trouble talking when my cheeks are pink. So I'll have to take a breath and then get started. I'm highly honored to be asked to come and to give you the Harrington Lecture and to be part of your 175th anniversary. Medical schools are the foundation of basic science research in our country. So I'm here to support all basic science research that's done. So I just wanted to mention that I had a typical and average upbringing. I went to public school, I went to college and got a bachelor's degree. I was bored, so I did some research and loved it and got a master's at the same time. I then went off and did an MD PhD at Boston University, did a medicine residency, went to the NIH, where I was actually able to get into Tony Fauci's lab and spent 7 years working in his lab, which was a great way to meet people from all over the world, as Tony had 125 people in his lab from the entire world. And I still work with many of them. After that, I was hired by the University of Pennsylvania and started my own lab and started doing research. So I'm often asked, what are the most important attributes of a researcher? And my response is always, well, there isn't a particular personality. You don't have to be an introvert. You don't have to be a particular type of person to be a researcher. But the one thing that I do believe is you have to be curious and you have to be creative. That's what basic science research is. But the interesting thing is that, and I put this slide up to highlight the point that curiosity alone is not an issue. If it was, we would have cats owning every major company in the world. And cats are pretty introverted and antisocial, so I'm not quite sure how that would work. They might end up in government, which actually could be a good thing. I haven't really thought through this, to have a bunch of introverted cats who worked instead of campaigned. But we can think about that later and maybe put it into a book. But the other important thing, along with curiosity and creativity, is intelligence. And you don't have to be the smartest person in the room or the world or the university, but you need a level of intelligence to be able to take the creativity and develop new things and figure out how to turn those into true therapeutics. When you think about it, research is really a game of whack-a-mole. So you're bright enough, you're creative. What happens then is new ideas keep popping into your mind. And the question is, what do you do with these new ideas? How do you decide which ideas are good and deserve to be followed up on? And which ideas are crazy or misguided? This is something that I have a lot of difficulty with, especially with the students in my lab. And the problem is some students will come in and say, I have a project that I want to do. And I always say, fine, let's think through it. Let's start doing the research. And sometimes it's a great project, and sometimes it's not. And then I have to sit down to them and say, look, I don't think this project is going to get you anywhere. I don't think you're going to get your PhD. I don't think you're going to be able to follow it. I just don't think it's a good project. And then they, of course, come back to me and says, well, wait a minute, you spent 25 years studying RNA with no success at all, no grants, no funding. But I say to them, well, but it's different, because I could see in the RNA therapeutics enormous potential for the treatment of many diseases. Unfortunately, your project doesn't have that, and you need to stop it, and you need to try something else. So science is a game of whack-a-mole. It's finding the right project and hitting it. I put this slide up because I was in Madrid earlier this year in an old Baroque church. And what it taught me is that the whack-a-mole game was actually invented a long time ago. And in fact, it's actually the mole that's whacking the people in this game. So we've updated the game because people probably wouldn't want to play whack-a-human, but that's how it started. So when you think about mRNA therapeutics, this is the basic timeline. And I always laugh because when I talk to lay audiences, I always get questions are, oh, I'm afraid of this vaccine because it was invented in 10 months. And I have to say, well, that's not exactly true. mRNA was discovered in 1961. So that's long before everybody in the audience was born. The first time it was injected, the audience that I'm talking to, not this audience. The first time it was injected into an animal was in 1990. And that was followed by a lot of collaborative research efforts. The work that Katie and I did fell into this region between about 2004 and 2020. But as you can see, it's not a simple step. It isn't somebody found RNA, they stuck it into an animal and made a vaccine and they were done. There was hundreds and hundreds of people and thousands of experiments that people did together to develop mRNA therapeutics. I just wanted to go over who the important people are in the direct production of this mRNA vaccine. Because Katie and I get a lot of awards for this, but we're not the only people involved. And there were many more before and after us. Peter Cullis and Ian McLachlan actually developed lipid nanoparticles. They developed them for siRNA, those are short interfering RNAs that knock out gene expression. Their interest was using them and they put them into clinical trials and it's now an FDA-approved drug. Soon after that, Katie and I discovered that we could modify RNA by changing the nucleosides to get rid of its inflammatory potential. The reason we did that is when we started to inject RNA into mice, using it as a therapeutic or a vaccine, the mice got sick and the mice died. And obviously you can't make a therapeutic where the drug kills the patient. So we spent 7, 8 years trying to figure out why RNA was inflammatory. After that, Norbert Pardee, and this is Katie left the lab and went to BioNTech, we figured out that the LNPs that Peter developed could actually deliver mRNA. And then Norby and I were the first to publish that you could make a vaccine with nucleoside modified mRNA and lipid nanoparticles, and it was incredibly potent. We did this during the Zika pandemic, and we showed that a single immunization protected macaques against challenge with Zika virus. After that, Barney Graham and Jason McClellan worked on improving the immunogen. So what's important to know about this is that immunogens of viruses are usually the fusion protein. So that's the protein that allows the virus to inject into and infect the cell. And for most viral diseases, you make the immune response against that fusion protein. What Barney and Jason and his group did is they figured out that most vaccines that were being made, and this is starting with the RSV, use the post-fusion form. So the post-fusion form is on the left. That's what the protein looks like after it's done its fusion. The problem is, you can see it's very different from the pre-fusion form. So the antibodies that block the post-fusion form don't stop infection. They don't bind the pre-fusion form. So the work that Barney and Jason did was to develop a spike protein from coronavirus that made a good immune response. And that's been incorporated into many of the vaccines currently in development. After that, this is just a small number of the researchers and pharmaceutical workers who developed the RNA vaccine for GMP production, who put it through clinical trials, and who made it for human use and for human distribution. So there's a huge number of people, and this is just the direct steps of making the COVID-19 vaccine. There were thousands of others before and after that have continued to improve it and to develop it further. I'm putting this slide up because Katie and I worked for 7 years until we figured out modified RNA. And we would go and we would talk to audiences. And this include friends and very important researchers like Gary Nabel and Tony Fauci and others. And we would say to them, we just made this great new RNA invention. And we would spend 20 minutes telling them about it and describing what it was. And they would say, oh, well, that's very nice, but why are you wasting your time with RNA? Pharmaceutical companies could care less. Biotech companies have no interest in it. It had failed clinical trials. It had failed all sorts of development. And we would say, well, but you have to think about what it's going to do and what it's going to replace. And what it's going to replace is protein therapeutics. And protein therapeutics uses 50,000 liter drums full of CHO cells to make the protein. And you have to reinvent the procedure every time you want to make a new protein. What that leads to is the cost of a monoclonal antibody therapy starts at $1,000 a dose and typically costs $50,000 to $100,000 a year. And our idea was RNA could replace all of that and do it much better. Because with RNA, a single reaction makes the RNA. It doesn't matter what protein is encoded for. A single reaction purifies it. You can make a new RNA in a day just by putting in a new template. It's very easy to scale up. It's very easy to expand, to deliver to patients. And as we know, it's safe and effective. So I just wanted to now talk to you about what Katie and I went through when we were trying to develop mRNA. So we started working together around 1998. And the story you've all heard is we met at a photocopy machine, and that's true. Back in those days, the only way you could read a journal article is if you photocopied the journal. And we both read a lot, and we both fought over the copy machine. So we started talking, and she says, I make RNA and put it into any cell. And I said, well, I'm a dendritic cell immunologist, and I'm interested in vaccines and therapeutics. So we started working together. The first grant we received was in 2007. And this was kind of a quirky NIH grant known as an STTR, which is actually a small business grant. And we thought we had a better chance because we could convince them that a small business and RNA might work well together. That was the first grant. So that's almost 10 years of work without any funding. And the way that happened is that Katie and I would work side by side in the lab. We didn't have any technicians or postdocs or anything who was helping us do this. It was all just Katie and I. She would make the RNA, I would add it to cells or give it to mice. We would sit and discuss the results. After that, we started getting a couple of grants, but as you can see, there isn't much here. And it wasn't until 2019, 2020 that grants started to come a lot easier. And now I can sit back and say, hey, we're in great shape. But we went 15 years without much grant support. So what that brings up is perseverance. And I gave you the dictionary definition of perseverance. Well, maybe not. Yeah. So the dictionary definition of what perseverance is, and then my favorite quote from Winston Churchill about what perseverance actually is. And I know the school teachers in the audience probably feel like this many days of the week nowadays. So they certainly understand. I put this up just to show our moment of success when the first phase 3 clinical trials came out for mRNA vaccines, and they showed nearly 95% efficacy and incredible safety. So this was our shining moment that Katie and I were suddenly recognized for what we had spent 25 years working on. I wanted to talk to you now about what's next, because that's what interests me the most. talking about the past is great, but I don't have a great memory, so I quickly forget my past. My wife always yells at me about that, but nothing I can do about it. But I'm interested in the future. And what the mRNA therapeutic landscape is useful for is that it's a platform, which means that it has an enormous number of potential uses. So we're developing vaccines for all of these infectious diseases and many others. We're developing vaccines for some of the more critical diseases in the world where previous vaccines have failed. Things like malaria, hepatitis C, HIV, and many others. But that's not all you can do with an RNA vaccine. So we're developing vaccines for food and allergen allergies. And any parent here who has a child with a peanut allergy knows that their life is a nightmare, that they're in constant fear when their child goes to school or goes out with friends, that they might eat a peanut and end up in the ER and be very sick. So we think it's going to be possible to make a vaccine that prevents peanut allergies and prevents house dust mite and tree pollen and weeds and cats and dogs and probably many different allergens. We're working on vaccines for autoimmune diseases. So currently autoimmune diseases are treated with immunosuppressive drugs, and they can sometimes be very immunosuppressive. We and others have shown that people being treated for autoimmune diseases, about 50% make a vaccine response when you give them a COVID vaccine. And that's, of course, a big problem. We're also making therapeutics for cancer and other types of vaccines. The more interesting thing, at least to me now, and I have, my mind is constantly going, so I'm always jumping to the next thing. You'll have to excuse that. But is mRNA therapeutics. And mRNA therapeutics is the delivery of a protein to a cell of interest. The big difference is that with RNA, you can replace intracellular proteins. So these are proteins inside the cell. Things like the CFTR gene, which is the defective gene in cystic fibrosis, and many other genetic deficiencies. We can deliver gene editing technology to fix broken genes. And I'll talk about a little of those next. Years ago, we showed that we could deliver mRNA LNPs to the brain. And we used it to treat an inflammatory disorder of the brain. So currently, the treatment of strokes is very limited. You can try anticoagulation therapy in a subset of patients. It works in some, but it's not, that simply modulates and helps to clear the vessels and reduce the inflammation. We figured out that we can deliver to the brain, and we can deliver a protein called thrombomodulin that's an anti-inflammatory, anticoagulation molecule, and we could suppress the inflammatory effects of strokes in animals. This is now being taken forward into further trials. I have to mention CAR T cells. So CAR T cells were developed by St. Jude's, but Penn took them into clinical development and got the 2 FDA-approved drugs. What a CAR T cell is and how it differs from a regular T cell. A regular T cell sees a little piece of a protein, a peptide of 9 to 11 amino acids. And that's determined by the MHC. So every T cell in every person is different. What a CAR T cell does is they stick a piece of an antibody onto a CD8 killer T cell, and they direct it to a tumor antigen. It's turned out to be an incredibly effective therapy for what we call liquid tumors, leukemias and lymphomas, huge effects of increasing survival. The problem is, this is what it takes to make a CAR T cell. You have to leukapheresis the patient. So what that is they that they put a big catheter into your vein and they run your blood through a machine for about 2 hours that takes 2 billion cells out of your body. Not everybody can tolerate that. It's not a simple procedure. You take those 2 billion cells and you infect them with a lentivirus to put the CAR gene into the cells. You then stimulate those T cells for about 10 days to make many of them, to make 1 trillion of them, and then you give them back to the patient. And that's incredibly effective for cancer. The cost of one treatment is about $475,000. And that's because it's 2 weeks of laboratory work under GMP, protective conditions, et cetera. What we've done is we figured out how to make CAR T cells in the body. So what that involves is we take a lipid nanoparticle and target it to T cells. And then we put a CAR molecule as the mRNA. So when the T cell binds an LNP, the RNA is released, it synthesizes and makes a CAR molecule on the surface. And that CAR T cell can kill tumor cells or treat fibrosis or a variety of other things. So in conclusion, I talked about all the vaccines that we're developing. I think mRNA therapeutics in the future is going to be even wider and have many more diseases that it can treat. We're looking at treating sickle cell anemia. So sickle cell anemia is a genetic disorder of the beta hemoglobin gene. It's primarily seen in Africans and Indian populations. About 200,000 people a year are born with sickle cell anemia. In the US, there's a treatment. We can do a gene therapy where we take a large amount of bone marrow out of a patient, infect it with a lentivirus, and give it back. Can you imagine doing 200,000 bone marrow treatments a year in Africa and India? The cost, the infrastructure would kill us. What we're going to do is we can give a single injection of LNPs targeted to the bone marrow, deliver gene editing technology to fix the broken gene. So now it's a single injection, and that's easy to do in any part of the world. That can be expanded to the hundreds of other genetic disorders of the bone marrow, or genetic disorders of the liver or of other organs. It can be used to deliver therapeutic proteins like antibodies and immunosuppressives. I used to joke with Katie, we would sit and keep making lists of everything we could possibly do. And it went on for pages and pages. And it's just so much, it's unbelievable. So I need to thank all of the people in my lab and all of the people in the labs that I've collaborated with on the work that I've shown you. And I'll stop there. Thank you.
Thank you for that wonderful talk. And I'm excited to delve in a little bit deeper with some of my colleagues this afternoon. My name is Jennifer Surtees. I'm an associate professor in the Department of Biochemistry here in the Jacobs School. My research is actually focused on DNA repair pathways and implications for cancer and neurodegenerative diseases. But through the pandemic, from the very beginning of the pandemic, I was fortunate enough to leverage resources here at UB to assemble a team to actually sequence SARS-CoV-2 from patient samples throughout Western New York, in collaboration with our healthcare partners across the county. And so when you hear about variants of concern in Western New York, know that that information most likely came from UB. I am pleased to welcome 3 of my esteemed colleagues here who are going to join us on the stage. First is Dr. Tom Russo. Dr. Russo is a SUNY Distinguished Professor and Chief of Infectious Diseases in the Department of Medicine. He has an active nationally funded translational research program that focuses on gram-negative bacilli. Dr. Gabriela Popescu is a professor of biochemistry and a leading neuroscientist who directs NIH-funded research on the biophysical mechanisms of learning and memory. And Dr. Jonathan Lavelle is a SUNY Empire Innovation Associate Professor in the Department of Biomedical Engineering. His research focuses on developing novel nanomedicine approaches to meeting unmet needs in treating, diagnosing, and preventing disease, including COVID-19. So I'm thrilled to have my colleagues here with us, and we're going to have a little conversation with Dr. Weissman.
Can you hear me in the back? Cheap seats? Good. So I think a lot of people in the audience would love to know the answer to this one. A pan-coronavirus vaccine active against all known variants and those yet to come is the holy grail, at least for COVID. Is your lab working on this? Do you think such a vaccine can be developed? And if so, how long will it take?
So it's a great question. I have to start off with a story that most researchers here will probably know. In spring of 2020, we started working on a pan-coronavirus vaccine. And I did this in collaboration with Duke and UNC and UCal and a few other universities. We submitted a grant to the NIH and they reviewed it. And some of the reviewers said, this is silly, we don't need a pan-coronavirus vaccine. And they rejected our grant and didn't fund it. So while now we all think it would be a great idea to make a pan-coronavirus vaccine, that wasn't always the case. So we've been working on this for approaching 2 years, or over 2 years actually. We've made a number of immunogens that we've published that work. So what you have to know about the coronaviruses is that there's different families. There's alpha coronaviruses, and then there's 2A, 2B, 2C, and 2D families. COVID-19 is 2B. MERS is 2C. The common cold coronaviruses are 2A and A alpha coronaviruses. We've made an immunogen that works for the 2B family, SARS and COVID-19. It protects against variants, it protects against bat-transmitted coronaviruses. We're working on 2C, 2A, and alpha variants, immunogens, and we hope to have those made within the next year and start clinical trials within about 2 years.
It is widely accepted that the mRNA technology is very successful and safe. And you've already told us how it can be used for COVID-19. And you've touched on how it's going to be used beyond that. Can you expand on it? Do you envision a library of vaccines waiting for the next pandemic? And how about all the other diseases you talked about, anemia and cancer? And how do you see that being used for that?
So actually, we had 5 phase 1 clinical trials that we started before the pandemic hit. So 2 were for HIV, 2 for influenza, 1 a universal influenza, 1 for genital herpes. That were phase 1 clinical trials that were in process. We were making the RNA and getting the patients lined up. They've all slowed down a little because of the pandemic. But since then, we've added a number of new vaccines that we've developed to clinical trials, including norovirus, C. difficile, malaria, and we're working on a few others. So I think the RNA platform as a vaccine has enormous potential and is in clinical development already, which means within the next 5 years, hopefully, we may see these in patients. The other therapies, So a company called Intellia has already done a phase 1 trial delivering modified RNA to knock out a gene called HTTR. It's a transthyretin amyloidosis, which is a disorder where people die within about 5 years of diagnosis. They did a phase 1 trial, and every patient that got the high dose of the drug has been cured for over a year. So it's already shown that it works for certain diseases. We're hoping to have our sickle cell anemia therapy in patients within about 2 years. And after that, the expansion is just enormous. So, a lot of this is how quickly we can get money to do the studies and convince the FDA to do the trials.
So just to sort of follow along that idea, one of the concerns with the mRNA vaccine the mRNA technology is having the infrastructure in place around the world to implement it in beyond the United States and Western countries. I wonder if you could speak a little bit about the challenges there.
Yeah, so this is something that I've been working on my entire career. When I was a fellow at the NIH, I set up research in Thailand. When I first started at Penn, I set up a research lab in Botswana. So I've always been interested and committed to access to therapies around the world. I've been working with researchers in Thailand for about 6 years. And it started through a friendship that I made at NIH. And we started talking, and I've been working with them to develop RNA vaccines and RNA therapeutics. In the early spring of 2020, Kiat, my friend in Thailand, called me and said, I've talked to the government, and we're nervous that any vaccine made in the West won't get to this part of the world for years. And we're not willing to stay shut down for years waiting for a vaccine. And the government was willing to fund making a vaccine in Thailand that's available to all of Southeast Asia and making a GMP production site. So GMP are the conditions that vaccines and drugs are made under. And they're very expensive, highly specialized conditions. So we started to make a vaccine in Thailand and produce a GMP site in Thailand that's now up and running. They're in the middle of a phase 3 trial and hope to give drug to their populations and in those of all of Southeast Asia within the next few months. We've built a GMP site in South Africa that's currently up and running and making GMP drug. We're building one in Rwanda. We're talking to Brazil and to Kazakhstan and a few other countries looking to build GMP sites there as well. My idea is that having access to being able to produce vaccines is critical. And a lot of it is many diseases pharmaceutical companies have little interest in. Dengue is a widespread disease, but mostly in low and middle income countries. One dengue vaccine has been made, it doesn't work very well. There's little interest in making another. Malaria is another vaccine that's critical for production, but there's no money to be made in a malaria vaccine, so pharmaceutical companies have no interest. There are many diseases that are limited to regions where pharmaceutical companies have no interest. So if we have GMP production sites and we have researchers who are making those vaccines and investigating those diseases, the local countries, local researchers, local companies can get together and make the vaccines themselves. And they may only just be distributed in those surrounding countries, but that's fine, that they have the ability to do it. And that's what's important.
So that's really exciting. And I have to say, one of my sisters lives in Bangkok, and she and her family struggled to get vaccinated actually because of the lack of access there. And I remember her telling me when they were actually starting to do exactly what you talked about and develop their own. So that's really cool to hear that you were involved in that as well. Jonathan.
Thank you, Dr. Weissman, for such a great talk and all the inspiration that's provided. My question is, practically speaking, what were some of the ways in which you were able to sustain this mRNA technology in the face of some of this, significant skepticism you were describing? And the second part is, what sustained your certainty that this was going to be worth pushing for? Were there colleagues that you mentioned or other sources of encouragement that helped you along the way?
Yes, so it was really Katie Carrico and I, and we sustained each other. And we both saw the importance or the potential importance of mRNA. I have to be careful how I describe how we sustained ourselves. When I moved to Penn, I got a lot of money as a startup package, and then got a bunch of NIH grants. So that startup package was extra money. And I convinced our dean to let me keep it. Usually they take it back after 5 years, but I was able to keep it. And that's how I supported Katie and I doing the RNA research for 10, 15 years until we got our own grants.
Right, so this is how the sausage gets made, how you sort of scrounge around and try to find the resources to keep you going, and hopefully have supportive deans to do this kind of thing as well. It's great. So I wanted to ask you a little bit about, you've had a lot of attention recently, less attention before that, but I'm curious about how this attention has made you think about how you talk about your science and how it has maybe changed how you communicate with the general public and how you've thought about how to communicate with the general public, and particularly thinking about trust in science and understanding how science works.
Yes, so my family and my friends will tell you I'm incredibly uncomfortable sitting up on this stage. And I do not like to present in audiences. I don't like attention. I'm happiest when I'm sitting in my lab working. But I've had to learn how to be in front of audiences and to talk to people. Something that I started very early on in the pandemic was talking to lay audiences. So I would spend many evenings and weekends talking to lay audiences about what coronavirus is. And then when the vaccines were released, I started to talk to them about the vaccine and why you needed to take it and why it was safe. And I've heard every crazy reason why people don't want the vaccines. My favorites are, somebody said, well, I heard it makes you magnetic. And what I thought in my mind is, well, actually, that's a good thing. I won't be able to lose my keys anymore. But I couldn't say that to them. So I learned how to talk to lay audiences and explain to them what a vaccine is, how it works, and why it's safe. And the critical thing that I repeated over and over is that 1 in 200 people die of COVID. You don't die from the vaccine. Can you see the connection between those two things? And if a person could see that connection, they would take the vaccine. There are groups of people who just don't believe in science and think that scientists are lying to them. Those people I've never figured out how to communicate with. They don't want to talk to me. They send me death threats. So I haven't solved the problem. I don't know how to reach large groups of people who don't believe in science and have made up their mind based on social media. But when for hesitant people, I hope that I'm able to convince some of them to take the vaccine.
Yeah, I mean, I think that I think that something as a responsible scientist, learning how to do that is actually part of our job in terms of communicating this kind of information. Yeah, I want to actually ask my colleagues up here if they have any other questions that they'd like to ask.
I have a lot of questions, but I'm just impressed and very grateful for how appreciative you were of you still have the slide with all the people that actually worked on it. What can you say about your own lab over the past 20 years?
So, I mean, I run the lab, and what that means is I get the money and I help with directing, but they're the ones who do the work. And many of the ideas come from them, that they don't all come from me. And I learned that when I worked with Tony Fauci. Tony set the environment. He gave us the lab, he gave us people, but the lab functioned on ideas that we came up with and that Tony helped to modulate and improve. So nothing is, we're not a dictatorial company where I say, do this, do this, do this. It's completely interactive and everybody in the lab contributes. Everybody in the lab has ideas and they're all incredibly important in getting all of this done.
Jonathan?
I was just wondering, were you at all surprised when you saw that moment of gratification or redemption, or that you were mentioning when you saw the phase 3 trial results? Did that exceed your expectations?
So I was mostly panicked because, so we had developed probably about 20 different RNA vaccines for a bunch of different diseases. In animal models, just about every one of them gave 100% protection. Influenza, Zika, herpes, they all were perfect. And we had never seen a vaccine that potent before. When I made the first influenza vaccine, I gave the sera to an influenza collaborator to test. It took him a month. And he came back screaming at me that I gave him spike samples because he had never seen a response that potent. So I knew that the vaccine worked well. The biggest fear of a basic scientist is everything works in mice. The statement is mice lie and macaques exaggerate. So the big fear is always when you get into people, isn't going to work. And that was my panic because I knew it was a great vaccine, but I didn't know if it was going to work in people. And when I saw 95%, I said, well, it could have been 100, but I'll accept 95. That's pretty good.
People aren't mice.
Yeah.
Sure, I just have a quick question. So you touched upon for the audience the various critical parts of the RNA vaccine, the lipid nanoparticle, the stabilized RNA itself with the substitutions, and the actual process of making the protein as well. This is version 1.0. Obviously, there's room for improvement. What of those steps, or perhaps something I didn't mention, do you envision has the greatest room for improvement moving forward to really develop even better vaccines than we have right now?
So Katie always gives me a hard time when I say this, but I think the RNA has the least room for improvement because we did such a great job making it. But I'm sure there's plenty of room for improvement. The LNPs with RNA is new. That's only been around for years. We've already made LNPs that are 5 times more potent. So I think better LNPs are going to make the vaccine more potent and reduce the sore arm and flu-like syndrome that people get. I think immunogen development is critical. So for things like coronaviruses and influenza, we've got a lot of knowledge. For things like HIV, hepatitis C, and malaria, we're just in the beginning of figuring out how to make an antigen that induces the right kind of response. There have been 50 HIV vaccines in various clinical trials. None of them have induced neutralizing antibodies. One of them had 30% protection for a year that disappeared, that couldn't be repeated. So we're nowhere with an HIV vaccine. I think by improving the antigen in the vaccine, we might get somewhere. And I think that's going to be critical for many of the vaccines we're developing.
So this has been really interesting, and I'd like to close with a final question, which is, what sorts of lessons have we learned, or do you hope we have learned, or we could have learned in the United States in the pandemic in terms of how we do science and how we work together?
So I think you have to divide that up. For the scientists have always been collaborative. This is nothing new. COVID-19 didn't suddenly make us much more collaborative, but it's highlighted the importance of collaboration. And the hope is that people continue and increase their collaborative activities. So I think from a science point of view, we were already in a good position. And the COVID pandemic has increased our collaborative nature. When you start to think about preparedness for the next pandemic, government responses, I think all of those need a lot of work. And I think we need to have more input of scientists into government, into public health, CDC function, in order to get those points across and to improve our preparedness for the next pandemic. And there will be more.
Well, thank you so much for discussing this further with you. I'd like to thank my fellow panelists, and thank you again for a wonderful discussion today.
Thanks to the four of you, and Dr. Weissman, if you wouldn't mind remaining on the stage for just a moment. You know, we've all heard brilliant people give talks, but I don't remember one more interesting and entertaining than this one. It was really, for a guy who likes to be in the lab, you do fine at the podium, real fine. And so now it's our honor to present you with this memento of the Harrington Lecture and your trip to Buffalo on your way to Geneva. It's a little bit fragile. It's very beautiful. If you get a chance to look at it, it's really gorgeous. So now after that really invigorating discussion, I'm sorry we didn't have time for the audience to ask questions, because I bet you anything you would have had some. At this point, what I'd like to do is ask our Distinguished Alumni Award recipients to join me on the stage, Dr. Robert Gore, Jessica Williams, and Kenneth Gales. And also, I would like to ask Dr. Raul Vasquez to join me at the podium. He will begin by presenting the 2022 Distinguished Alumni Award, and this is the Medical Alumni President's Award. Raul.
Welcome, everyone, and congratulations to all the awardees, and thanks to Dr. Weissman. As president of the Medical Alumni, it is my pleasure to introduce this year's Medical Alumni Associate President's Award, Dr. Kenneth Gales, alumni of class of 1973, for his role as a model mentor whose actions continue to serve as an inspiration for others. Dr. Gales was born and raised in Buffalo. He earned both his undergraduate and medical school from University of Buffalo. He received his postdoctorate training in cardiology through residencies in Michael Reese Hospital in Chicago, Illinois. Dr. Gales became the first African-American invasive cardiologist in Western New York. I want to reiterate first. And I think it's often time we don't get to see people that look like him recognized early in their life. He was a mentor to me, as he was to many students. He showed us compassion and he gave us the support we needed. While he was serving as medical director at Sheehan Hospital, Dr. Gales took the time to teach me the art of business and leadership, helping me to understand different models of care, especially within the hospital system. I felt a sense of belonging with him as a mentor and a friend, and have stayed in touch with him for over 25 years. His contribution always has been one of support, especially to the minority community. I dedicated this award to someone who encouraged me to thrive and feel a sense of belonging, which is so important to have someone who looks like me and practice in a community that looks like us. So again, thank you for everything all these years, Dr. Gales.
Can't let him go without giving the memorial plaque of his year as president of the Medical Alumni. Congratulations, Dr. Vasquez. It's now my privilege to present the Distinguished Medical Alumnus Award to Dr. Robert Gore, a 2002, I remember you, by the way. He was good then too. A 2002 graduate of our school who is celebrating his 20th year reunion. Doesn't seem like it's been that long. Most days, it doesn't seem that long. Dr. Gore is an attending physician and clinical associate professor at Kings County Hospital, SUNY Downstate, in the Department of Emergency Medicine in Brooklyn. He's the founder and executive director of KAVI, K-A-V-I, Kings Against Violence Initiative, an internationally recognized hospital, school, and community-based youth violence intervention, prevention, and empowerment program directed at teens and young adults. In 2018, Dr. Gore was named a top 10 CNN hero, a global honor that celebrates everyday people changing the world. That same year, he was also named to the class of Presidential Leadership Scholars. He and his anti-violence work with youth have been featured in the New York Times, History Channel's History Now series, and many other prominent media outlets. Born in Buffalo, raised in Brooklyn, Dr. Gore is a graduate of Morehouse College in Atlanta. He earned his medical degree right here at the Jacobs School in 2002 and completed his residency in emergency medicine in Chicago at Cook County Hospital. Congratulations, Dr. Gore. So good to see you here. Love the pants. Thank you. It's also my pleasure now to introduce Dr. We lost it. There it's back. As this year's recipient of the Distinguished Biomedical Alumnus Award. Dr. Reynolds earned a doctorate in pathology in UB in 2004 and today serves as associate professor in the Department of Medicine. Her innovative research focuses on the development of therapeutic delivery tools using nanomedicine to modulate both the nervous and immune systems. We recognize Dr. Reynolds for her excellence and expertise in nanomedicine for cancer, as well as for her research focused on HIV, TB, and other infectious diseases, including her recent studies on the testing implications of COVID-19. In addition, she serves as vice chair for faculty development in the Department of Medicine, and that entails leading all faculty mentorship, career guidance, and faculty promotion efforts. Congratulations, Dr. Reynolds. Thank you to everyone. And before we leave, and this is not scripted, by the way, I'm taking, when you give me the podium, you run the risk of something happening here. This is also the 175th anniversary of the AMA. And I want to share with you some comments that were made by a really distinguished man, now deceased, at an AMA meeting 23 years ago. His name was Rabbi Herman Schallman, and people are still talking about what he said 23 years ago. And so while I'm not of his faith tradition, his message is so universal and so appropriate for a day when physicians and scientists who have trained here and have devoted their lives to improving humanity, and certainly, Dr. Weissman, the number of millions of people whose lives have been saved by the work that you and your colleagues started all those years ago. So if you would allow me, I would like to just share what Rabbi Schallman said and apply it to the people in this room. When I look out at this assembly of men and women, I am overwhelmed by the realization of how many thousands, nay, perhaps tens of thousands of lives who have been saved by you, how much pain has been alleviated, how much despair defeated, how much hope you have given to those who were ailing and to their families and friends. Today, I can say with our ancestor Jacob, this is a house of God, and I did not know it. I stand on holy ground. So thank you all very much. And now we get to the fun part. We're going to get, let's see, those of you who are celebrating your anniversaries and reunions, please join us in the atrium, have a beverage, take some tours of the school, and as well as stay for dinner tonight. Our tour guides are near the elevators, ready to escort you. Golden reunion groups, please join us just outside the lecture hall by the piano. Did I forget anything, Jennifer? Great. Welcome, everybody. Thank you for joining us. Congratulations to all our recipients, and Dr. Weissman, unbelievable. Thank you so much.